In lecture video B, we're going to cover slides 11 through 14. We are going to look in detail at transcription. In part C, we will look at translation. I choose to separate transcription from translation because this tends to be a challenging piece of information for students. Both of these processes are broken up into initiation, elongation, and then termination, which that is how it works. But it oftentimes can be confusing for students. So we're going to look at transcription and this process in a separate video to kind of help you digest these two processes as separate pieces of information. All right, without further ado, let's come back down here. And we're going to look at transcription in detail, how we take DNA and use it to make messenger RNA. So the process as a whole of gene transcription in bacteria happens at the exact same time as DNA replication because there is no nuclear membrane as a barrier between any of this stuff. It can all happen at the same time, which is one of the reasons that bacteria can grow so much faster than any other organism. It's simply because they can undergo these processes by having them kind of overlap. They're all going to happen at the same time. All right, so we're going to start with transcription, which is the first part of gene expression. This is where RNA polymerase is going to read the DNA and it's going to make messenger RNA according to base pairing rules. We call that messenger RNA molecule that is made a transcript. So that's why this is called transcription. In bacteria, the vast majority of their genes are going to be transcribed and ultimately translated into a protein. So we say that most of their genes encode proteins. They are going to be used to make a functioning protein. When you contrast that with like humans, for instance, we have a whole lot more DNA, but we have a lot of DNA that won't ever be, in fact, the vast majority of our DNA won't ever be used to make a protein. Some of it is regulatory for our cells need, but a good majority of it is just kind of junk DNA. It's not necessary and it's not going to be used. But bacteria are optimized for their environment and they have only the genes that they need. So the genes that they have, they're going to be using. They're going to be using them to make proteins. So another kind of unique setup to maximize efficiency in our bacteria are sets of genes that are going to work together. So we say that they work in tandem. They work with one another. Oftentimes we'll have multiple genes, each for, say, an enzyme for a metabolic pathway that are going to be encoded together. That collective group of genes is going to be controlled by one specific regulatory part. Each gene is going to have a promoter. That's the beginning part. Well, in bacteria, their genes in what is called an operon have one promoter that is going to control multiple genes within that operon as a whole. We'll talk more about that here in section C, lecture video C. But it's important to know that this is another way to optimize your energy usage. So by controlling all genes with one promoter in that operon, we can turn all of those genes on collectively. But if we don't need their proteins, we can turn those genes off. We'll look more at that in detail later. So here, as we begin to move into the details of messenger RNA and how it is made, I want to clarify that mRNA is still RNA. It's going to consist of A's, U's, C's, and G's. It is going to be single-stranded. But there is a different kind of RNA called transfer RNA. tRNA, transfer RNA, is going to become kind of a central part of translation when we talk about it. Transfer RNA is going to transfer in amino acids to our ribosomes. So just to clarify, it's important that you understand in this part of the video, we're going to be dealing with messenger RNA. How do we make it? All right, so when we look at DNA, DNA is double-stranded. One of those two strands is going to be utilized in making messenger RNA. The other strand will not be used in making that messenger RNA. So the strand of DNA that is used by RNA polymerase is called the template strand. The strand that is not used is the non-template strand. We aren't going to go into details about what makes the template strand different from the non-template, but just understand that the template strand of the DNA is going to be the one that RNA polymerase is going to use. So that's what we're depicting up here. We've got some important kind of things to point out. Again, this is one that I pulled from Google because it's nice and simple and I like it. So our two strands of DNA are there in blue. The template strand in this example is on the bottom. Note that there is a little red section of DNA on that template strand. That is called the promoter. This is going to be the start that RNA polymerase is going to be looking for. So RNA polymerase is an enzyme that consists of multiple subunits. It actually consists of 4 individual subunits. That enzyme has a 5th subunit that can sometimes be attached to it. This 5th subunit is called the sigma factor. When we have those 4 RNA polymerase subunits and then that 5th sigma factor bound, our RNA polymerase is in its kind of full form. It's ready to go to work. There are lots of different versions of what is considered to be a sigma factor. It just depends on the different type of species. But we are always going to have that sigma factor because it's going to help us identify that promoter region on our template strand of DNA. So when we have those 4 RNA polymerase subunits and that 5th sigma factor on there, we refer to that RNA polymerase enzyme as being a holoenzyme. So essentially it is intact. It has all of the parts that it needs to recognize the promoter and go to work, undergoing transcription of the gene. So RNA polymerase, its job is to read that template strand, and according to base pairing rules, RNA polymerase is going to make our messenger RNA. Let's look at the details of that. Remember, we've got initiation, elongation, and then termination. For transcription, we are elongating messenger RNA. That's what we are making. So when we look through this process, helping you to remember and identify what it is that you're elongating, what you're making, is going to be helpful for you. Okay, so we'll point out what it is that we elongate in translation later. So in initiation, okay, we've got our RNA polymerase with that sigma factor bound. So it's that holoenzyme. Its job is to really scan and check out that DNA. It is looking for the promoter sequence, which in this particular region we have denoted as red. Okay, so here is our RNA polymerase. We see that sigma factor bound. We're just we're bouncing along. We're scanning things, checking things out. When we get to that promoter, we are going to have our holoenzyme kind of clamp down on it. It's going to bind really tightly. And it's going to be kind of stuck at that promoter region. In order for our RNA polymerase to actually do any work of moving down our DNA and making that messenger RNA, the sigma factor has to fall off. So that is kind of one of the key steps before we move into elongation. We've got to get rid of that sigma factor. So we're going to have our holoenzyme come in, and it's going to find that promoter and clamp down. RNA polymerase is going to separate the template from the non-template strand. This is different than what helicase does. Okay, so helicase is not part of this. RNA polymerase is going to be the one that's going to separate the strand of DNA. And then it's going to look at the nucleotides that are on that template strand. And it's going to begin to pair those bases accordingly, bringing in A's, U's, C's, and G's according to the template strand. We have to have that sigma factor fall off. So once that sigma factor falls off, we see that down here at the bottom, it leaves the complex. Now our RNA polymerase can move down our gene and make this messenger RNA, which they've depicted as a little blue line. So this is our A's, U's, C's, and G's right here. And it's going to make that according to the sequence of nucleotides in the template strand. And it's going to keep going until it reaches the end of that gene, what is called the termination region. So the terminator region of a gene signifies to RNA polymerase, hey, you're done. Thanks for coming. You made our messenger RNA. Great job. Once RNA polymerase reaches that termination region of the gene, it will fall off. We will release that messenger RNA. So we said at the beginning of this, though, that we don't waste any time getting to the second part of gene expression. So translation is going to start even before transcription has ended. So before we're even done making that messenger RNA, our ribosomes, and we're going to have multiple of them at the same time, they're all going to hop on. So here's one ribosome, here's another ribosome. We can have a whole bunch of ribosomes that are simultaneously translating that messenger RNA into a protein, okay? We will look more at what those ribosomes are actually doing here in lecture video C. But be aware that this is not like a discrete process. We don't do transcription and then we're done, and then we do translation and we're done. This all overlaps on itself. I'm going to show you the transcription animation that your textbook provides. Please be aware that there is more information in here than you need to know, but I want you to see RNA polymerase in action. I don't care that you understand the specific names of the RNA nucleotides. I also don't care that you know how energy factors into all of this.
The first step in gene expression is to make an RNA copy of a gene's DNA sequence. That is, to transcribe a gene. Transcription includes the stages of initiation, elongation, and termination, and is performed by an enzyme called RNA polymerase. In bacteria, before initiation, RNA polymerase includes a number of subunits, including one called sigma. Sigma factor binds tightly to promoters, which target the polymerase to a gene. With the sigma factor attached, the composite RNA polymerase is known as a holoenzyme, meaning that it includes all the subunits needed for initiation. The holoenzyme form of RNA polymerase binds loosely to any sequence of DNA. Thus, within the cell, the RNA polymerase binds and comes off DNA rapidly, as if scanning the sequence. When the RNA polymerase binds to a promoter sequence, however, the binding is tight. To start transcribing a gene, the RNA polymerase opens the DNA, creating a transcription bubble. Now the RNA polymerase can fit a ribonucleotide to base pair with the DNA. So begins the elongation stage of transcription. To elongate the chain, the polymerase catalyzes the addition of new ribonucleotide triphosphates, RNTPs. Each incoming RNTP condenses with the 3' hydroxyl of the preceding nucleotide, releasing pyrophosphate in a step that yields energy. Only a few ribonucleotides can be added before the polymerase stops, because the whole complex is still tightly bound to the promoter. However, now the sigma factor falls off the complex, allowing the core RNA polymerase to continue to move along the template, synthesizing RNA at approximately 45 bases per second. Finally, transcription must finish at the end of the gene, at a pause site in the DNA sequence that causes the RNA polymerase to fall off.
That pause site is the terminator.
In bacteria, even while transcription is occurring, the next stage in gene expression, the translation of a protein, also begins. Here RNA polymerase is transcribing a gene. The beginning of the growing mRNA has a region called a ribosome binding site, where a ribosome can bind and start translation. As the mRNA lengthens, additional ribosomes can hop on, creating a polysome, which is an mRNA with a series of ribosomes translating the sequence into proteins. This electron micrograph shows a polysome. Notice how the ribosomes farthest from the 5' end, the beginning of the mRNA, show the longest growing peptide chains.
All right, so there's a little more detail there than I expect you to know. But more than anything, I wanted you to see RNA polymerase in action. I wanted you to see its movement, how it finds that promoter and clamps down, how we begin to separate those two strands so that it can have access to that template strand, and the fact that the sigma factor must fall off before we can proceed all the way down. We keep going until we get to that terminator site, and then at the terminator site, the RNA polymerase knows it's done, and so everything will disassemble. Before you move on to translation, make sure you've got a good handle on what transcription is. This process is going to kind of come back, and we'll rehash it several times as this course proceeds. So make sure you understand the details of this one.